Mitosis allows for growth of the tapeworm and repair of tissues in a tapeworm. It maintains all the instructions in the original cell since the cells produced are identical. The tapeworm also changes forms in various hosts to exploit various environments. Mitosis makes these changes possible. Incorporation of HGH gene into tapeworm. A virus entered a human body cell and its DNA became incorporated into the human DNA. By chance, the viral DNA became incorporated close to the human gene for HGH. When the human DNA began replicating viral DNA, the HGA was replicated as well and became a part of the viral genome. The virus mutated in such a way as to allow it to enter the reproductive cells of a tapeworm gamete. Once inside a tapeworm cell, the viral genetic information became incorporated into the tapeworm DNA. The gene for HGH then became part of that tapeworm's DNA. Since it was gamete, this altered gene would be passed on to subsequent generations of tapeworms. Dwarfism and treatment. Lack of HGH during childhood development results in the disorder known as dwarfism. Children with dwarfism develop at a greatly reduced rate. However, body proportions are relatively normal. At puberty, children who lack HGH have long bones that are greatly reduced in length and hence they're dwarfs as adults. Advantages of using PGA instead of human HGA. Produces growth in a similar way to HGH. Does not cause insensitivity to insulin. No risk in leading to diabetes. Blood glucose would not elevate. Other diabetic symptoms would not occur. Any other reasonable response. Although there is a high level of glucose in the blood, in the absence of insulin, body cells are impermeable to glucose. Therefore, glucose is not available as an energy source and fatigue results. Since glucose cannot be used as an energy source, fat or protein stores are metabolized, which result in weight loss. Also, glucose is not stored, therefore weight gain is not possible. Glucose would be found in the urine of the patient with diabetes mellitus, or the urine produced by a patient with diabetes insipidus would be insipid, tasteless. The urine produced by a patient with diabetes mellitus would be sweet. Or the urine of the patient with diabetes mellitus could also have ketones present and could be slightly acidic or repellent. Thyroxine would decrease blood glucose levels by increasing cellular metabolism rates. Cortisol would increase blood sugar levels by converting amino acids or fatty acids or glycerol or fats or proteins to glucose. Epinephrine stimulates the conversion of glycogen to glucose by liver and muscle tissue, thereby increasing blood sugar level. Sensory neurons were damaged because the patient could not feel the pain caused by the broken ankle or lesions, but could still move their limbs, i.e. motor skill intact. Damage to or irregular functioning of blood vessels of the retina may result in permanent damage to the cells making up the retina, i.e. lack of O2 nutrient. If the rod or cone are damaged, then there will be reduced sensory reception and reduced impulses in neurons to the occipital lobe. This would result in some vision loss or even blindness. If the optic nerve is damaged by decreased blood flow, the sensory information is not passed to the occipital lobe of the brain. This would result in vision loss or blindness. Or, the increase in blood vessels may obstruct the light reception by retina cells, resulting in vision loss or blindness. Reduced sensory stimulation would occur, so the neurons would send a reduced impulse to the occipital lobe of the cerebrum. Evaluate whether conducting research would be useful for effective individuals. Possible advantages. An advantage of CH research for individuals is that the research might ultimately lead to a cure for CH. An advantage of CH research for individuals is that the research might lead to treatment for individuals with acquired or inherited illness. An advantage of CH research for individual is that the information gained about the nature of CH would be used in counseling individuals who have CH and are planning on having children. An advantage of CH research to society is that it may lead to a better understanding of the action of testosterone and estrogen. This increased knowledge could benefit society as a whole, potentially leading to health benefits for society. An advantage of CH research to society is that it may provide a better understanding of other X-linked traits. This increased knowledge could benefit society as a whole, potentially leading to health benefits for society. Other responses may be appropriate. Possible disadvantages. A disadvantage to a society of research to find a cure for CH is that it is very costly and may not be warranted for such a small population of affected individuals. A disadvantage to society of research to find a cure for CH is that it sends the message to society that if an individual does not appear normal, a cure must be found to rid society of a particular phenotype. Finding a cure does not promote the notion that society is at its best when made up of a mosaic of individuals. Other responses may be appropriate.